Hello, and we're glad you've joined us for today's live webinar, Genomics and Cytogenomics Microarray in CNS Neoplasia Case Studies to Consider. I'm Robert Castellanos of LabRoots, and I'll be the moderator for today. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. Today's event is also sponsored by Affymatrix. Affymatrix technologies enable multiplex and simultaneous analysis of biological systems at the cell, protein, and gene level, facilitating the rapid translation of benchtop research into clinical routine use for human health and wellness. Affymatrix, part of the Thermo Fisher Scientific, provides leadership and support, partnering with customers in pharmaceutical diagnostics and biotechnology companies and leading academic, government, and nonprofit research institutes in their quest to use biology for a better world. For more information, please visit affymatrix.com. So let's get started today. You can pose questions to the speaker during this presentation. And I just wanted to remind our audience today how to go about doing so. If you selected the green Q&A button at the lower left of the presentation window, you'll be able to submit your questions. All the questions shall be answered at the end of today's presentation. To enlarge the slide window, click on the screen icon at the lower right corner of the slide window. If you're experiencing any technical problems hearing this presentation, click on the support button found at the top right of the presentation window, or report your problem by selecting, again, the green Q&A button at the lower left. I would like to introduce today's speaker, Stewart Neill, MD, an Assistant Professor of Pathology and Laboratory Medicine and Associate's Director of Pathology Residency Program at Emory University. Dr. Neill is a neuropathologist at Emory. He received his MD degree from Emory University School of Medicine in Atlanta and completed a residency and neuropathology fellowship at Emory. I would like now to turn it over to Dr. Neill for his presentation.
All right, thank you very much. That was a very kind introduction, and I'm glad to be here with everyone. Unfortunately, we had a little bit of technical difficulty starting up, so I'm going to be a disembodied voice for you. You won't see me through a webcam, but I'm here to guide you through this presentation. We'll talk a little bit about my experience and our experience here at Emory doing some clinical genomic and cytogenomic workup in CNS neoplasia, and the main focus of this talk will be through using case studies to kind of illustrate some of the ways that we have highlighted important features of our cases through some of the genomic workups that we've done. So again, my name's Stewart Neill, and I'm an assistant professor in pathology here at Emory. And I've been at Emory for a very long time. I was an undergraduate here, did a medical degree here, and then my residency and fellowship here. Like being at Emory, but certainly enjoy interacting with you folks. And so if you do have questions, please feel free to ask them either now or at another time. So this is me. I don't have any conflicts other than to report that I do give some talks for Affymetrix from time to time in various locations. So again, glad to be here. Appreciate everybody's attendance and the kind invite from LabRoots and from Affymetrix to talk in this platform. So who am I really? So it's funny that I'm a neuropathologist really getting wrapped into this stuff sometimes. You know, obviously I trained in anatomic pathology, also in clinical pathology, but my main focus in my early residency training was that I would become an anatomic pathologist. But, you know, when I started in neuropathology fellowship, we really, at around the same time, we started getting into genomic workups of brain tumors here at Emory. And I can tell you, it was a transformative experience for me. It really changed the way that I approach my work, and has given me a lot of insight into how the tumors behave, how I can deliver better information for patients. So I'm very excited about this type of information and talking to you about it. So again, what are we going to do today? We're going to talk about our experience here at Emory and the use of genomic studies in the assessment of CNS neoplasia. We'll talk about some specific case studies, and then we'll also use those case studies as a way to discuss some of the investigative implications of the information. You basically can't do this type of work without running into things that maybe invite you to perform translational work as well. Try to carry some of your clinical information and gain insights into things that might implicate bench research or translational research. So first thing, just a quick introduction. So in surgical neuropathology, we're undergoing a real paradigm shift for how we think of what we do and how we do our work. And really this gentleman over here, this gentleman here is Dr. Harvey Cushing. He was a neurosurgeon about 100 years ago who really developed a lot of the he and the folks he worked with developed a lot of the terminology and histopathologic classification systems. The sort of infancy of our specialty was promulgated by him and the folks that he worked with. And a lot of the terms that we use today are really still based upon what Cushing described almost 100 years ago. But in the last few years, we have seen a lot of genomic studies be performed on brain tumors, both through things like the Cancer Genome Atlas and through other groups that have really fed back into how our diagnostic criteria are established and how we gain an idea as to prognostic and predictive information about the tumors that we deal with. So in the last few years, there's been we've convened a new WHO group to publish a WHO update that's just been put out in the last couple of months, basically changing the way that we do neuropathology diagnostics to incorporate genomic information. And so now when we issue neuropathology diagnoses, in the diagnostic line, we mention things like IDH mutations and 1p19q co-deletions, things that really bolster our diagnoses and make them both more accurate and precise. So in a lot of these new approaches have placed a lot of our previous notions in a different light. So now when we think about older cases, when cases are brought to us who maybe have been diagnosed 10, 15 years ago, and the patient is undergoing treatment here at our medical center, we oftentimes wonder, okay, really what was that tumor? And sometimes we end up going back and performing genomic studies using FFPE-based platforms to try to get an idea of, oh, what's the up-to-date diagnosis. So we really think a lot about all of our literature going back before, maybe 10 years ago, even more than 5 years ago, really is put in a different light based upon the information that we know now. So at Emory, briefly talking about our workup, of course, we still look at H&Es, and we do immunohistochemical studies on our tumors. The H&E is still the foundation upon which we base our later workup. We test for IDH1, ATRX, and P53 for infiltrating gliomas. On pediatric cases in particular, we will perform IHC for H3F3A K27 mutant protein. And then on medulloblastomas, we would perform GAB1, YAP1, beta-catenin, and P53 immunostains. On all gliomas, we would basically do a snapshot mutation panel, which is a multiplex PCR panel. Helps us with a bunch of different IDH1 mutations, the IDH2 mutations, BRAF mutations, and a few other ones. We'd like to adopt a larger NGS panel, but that's been in the works for a while now, and we're still working on it. We do perform MGMT promoter methylation analysis, and then we do OncoScan, our cytogenomic microarray, FFPE-based microarray that helps us in a number of different scenarios that we'll talk about over the course of today. So at Emory, we've performed OncoScan on a little over 400 neuropathology specimens over the last two plus years. The majority of those have been infiltrating gliomas, astrocytomas, oligodendrogliomas, glioblastomas. We've also done a bunch of other tumors, lower grade gliomas, ependymomas, what used to be called primitive neuroectodermal tumors now are called embryonal tumors, and some meningiomas. And I also mentioned that we've done an increasing number of weirdomas, what I call weirdomas. So odd tumors that perhaps would have generated a more descriptive diagnosis in the past, but that we've been able to use cytogenomic and genomic studies to sort of tease apart a little bit more about what they actually are and how they may actually behave. And in those scenarios, I mean, you know, you'd think they're rare, right? But in practice, we run into these on a actually relatively regular basis. So these oddball tumors, they give us opportunities to get a greater understanding of tumor biology, and we're working really outside of an incredibly high volume laboratory here. We get a moderate volume of specimens, so we really dig into a lot of the cases that we get. Not saying that folks at higher volume places don't do that, but we're able to take some time and really think about some of these oddball tumors that we get, and our genomic studies have helped us gain some ideas about what those things may mean. So just some general lessons that we've gotten performing this type of testing. We do prefer using FFPE material whenever feasible. This integrates well with our established workflows here in the histopathology laboratory, so forth. And it also allows us to select areas to test, to target, based upon what we see. So we can avoid areas with a lot of necrosis or cautery artifact. We can also, say in instances where there are areas of the tumor that look strikingly different, we can test those different areas just basically doing dissection, microdissection on unstained slides. So allows us also, with FFPE material, we can pull cases from the past and compare them to maybe to what we have right now. So in our experience, pre-analytic variables heavily affect the testing outcome. We use here unstained slides as our DNA source, and we review the corresponding H&E with the pathologist and therefore select the areas to test, and then that allows us to check for tumor presence and percentage and so forth. So we know exactly what we're testing. And we also make sure to make that the tissue is properly prepared. We avoid decalcified specimens. And we've also had problems in Oncoscan and slides that have been baked for IHC, basically have undergone antigen retrieval for IHC. Our microarray consensus conference is something that I want to highlight a little bit. So we get together every week. You get the anatomic pathologists, the molecular pathologists, cytogeneticists, and our technologists all meet weekly to discuss the cases. And that allows us, number one, to tackle anything that may have produced a problem case. Okay, we look through the pre-analytic sequences to get an idea of what may have been causing the problems in our cases. And then also, it allows us to all put our head together and agree on the interpretation of the case and put it in the context of the histopathology and clinical findings and so forth. So some more lessons here. Transmitting this data to pathologists and clinicians carries a lot of challenges because the different people in our brain tumor group have various different levels of exposure to the information that we're trying to transmit. So it puts a lot of emphasis on us, number one, getting on the same page amongst our group, and number two, transmitting that information in a very easy to understand method for our treating clinicians. And sometimes we use tumor boards as a platform in which to transmit that information. And then also, say we're doing some translational research, remember, analyzing older material may lead to an altered diagnosis, and so you need to be aware of that and also have an open dialogue with some of the treating clinicians if you are performing older studies that maybe you will discover something that you may not have expected. It's not uncommon for older cases to gain a fundamentally different diagnosis if you do genomic studies on them. So next we'll get into some of the case presentations. It's just selected a few cases to illustrate the correlation between histologic, genomic, and other clinical pathologic information. We'll talk about the implications of the diagnosis, prognosis, and therapy. And then we'll also explore some of the potential applications into the investigative realm from these cases. Okay? So our first case, This is a man in his mid-30s who presented to us with headache and nausea and vomiting, and was found to have a left temporal occipital mass. And so what you see here, and I'll go ahead and highlight a few things. What you see here are, okay, all these little sticks here in the background are axons and dendrites of the native brain that have been they've been joined by a bunch of cells like these guys with relatively rounded nuclei. Okay, and so this is a picture of an infiltrating glioma. Basically, all the little sticks in the background that you see are the native processes of the brain, and they are being infiltrated or invaded by a population of cells that is neoplastic. And so traditionally, we would tease apart infiltrating gliomas based upon those nuclear characteristics of the infiltrating cells. So we would say many of these nuclei look relatively round, so we would think maybe there is an oligodendroglioma component to this. But some of these cells also have plump pink cytoplasm, and some of the nuclei are a little bit elongated, okay? So maybe there's an astrocytoma component to it too. And so some people may have called this oligodendroglioma. Some people may have called it astrocytoma. Many people may have called it an oligoastrocytoma, which was fundamentally, in many cases, a wastebasket diagnosis where someone couldn't make the clear distinction of whether it was an oligo or an astro. So in the interim, we've delineated that using testing for IDH mutations and particularly 1p19q co-deletion, one can tell whether many infiltrating gliomas are oligodendrogliomas or astrocytomas. If you have an IDH mutant tumor, it is likely to be an IDH mutant astrocytoma or an oligodendroglioma, and you can use 1p19q co-deletion testing to make that distinction. The 1p19q positive tumors are, in this scenario with an IDH mutation, oligodendrogliomas, and those that are 1p19q negative fall mainly into a category of IDH mutant astrocytoma. And you can see the prognosis is different between these tumor groups. In fact, it's stated here on the slide 8 years and 5 years, but we've seen people with oligodendrogliomas survive much, much longer than that. And some people with infiltrating astrocytomas that are IDH mutant will survive a little bit longer than 5 years too. So we have, there is definitely a difference in prognosis between those two groups. And then importantly, if you have an IDH wild-type tumor, there are a bunch of different subgroups of that, but many of them behave incredibly poorly, very much like the old diagnosis of primary glioblastoma, and that has a very dire prognosis, 12 to 18 months. So it's important to make these distinctions. It really gives people a much clearer idea of what the tumor's prognosis is likely to be. So here's our carrier view of this case, and what you can see is that there are, so remember, blue is gain here, and red is loss, and these areas that are purple are LOH, okay? So what you see, lots of blue bars, all right? So that's lots of gains, and actually reflects a polysomy in this case. There is a detected IDH1 mutation here. But importantly, there's no, looking at 1p and 19q, there's no whole arm co-deletion, and so it most likely falls into the category of an IDH mutant astrocytoma. And then you can look here, and it actually has an LOH on 17p where TP53 is located. That also goes along with IDH mutant astrocytoma. So here, this is more of a general case, but we found a polyploid tumor that was IDH1 mutant, negative for 1p19q co-deletion, had an LOH event on 17p. And so our integrated diagnosis here would be anaplastic astrocytoma, WHO grade 3, IDH mutant. So the implications here, based on this histology, as I was mentioning, this tumor could be called, under the old classification system, a number of different tumor types. And so the problem there becomes patients travel with these patients who have these brain tumors. They tend to travel to different institutions. They want to get the best care they can. Maybe they want to get it on a clinical trial. And so those cases would be reviewed and perhaps given a completely different diagnosis at a different institution. And so, these patients are put into a terrible scenario of, well, which one do we choose? And the clinicians are confused, and it would make a big problem in the past for disagreement between institutions, disagreement between pathologists, and the patients caught in the middle. So based upon this genomic information, though, we feel like we can get a much clearer diagnosis that's more easily transferable between institutions. And so we've also run across some similar instances where tumors that had features suggestive of a wildly different tumor type, say a ganglioglioma, also came out with genomic studies very similar to this. And so we've not only been able to look at tumors where we would sort of expect this type of differential diagnosis, but tumors that, well, really didn't look like an IDH mutant astrocytoma or an oligodendroglioma, where the genomics really points you in a particular direction. So our integrated classification offers greater accuracy and precision moving forward. And we have to remember, what does this imply for the tumors that we've diagnosed in the past, or the ones that are stored in tumor banks? This is a scenario that's going to come up on a regular basis, not just in neuropathology, but as we apply this type of information in other solid tumor types. So our second case. Similar looking thing here, okay? So we start out here. We see this is a woman in her mid-40s who came with headache, nausea, and vomiting, was found to have a right frontal interparenchymal mass. And here we see, again, our background neuropil, our axons and dendrites are splayed apart and are infiltrated by round to oblong nuclei of an infiltrating glioma. And so our carrier view here again shows no 1p19q co-deletion, okay, and also detected a mutation of TP53 on 17p. Now, I'll tell you that with OncoScan, we don't always trust the detection of TP53 mutations with OncoScan. Now, the IDH1 mutations, we've had a lot of luck confirming those, so we certainly trust the assay's detection of IDH1. TP53 is a little bit, it's been a little bit harder for us to confirm in some scenarios. So, but we also see some other things with this carrier view. There are some other abnormalities. Look over here on 11. That looks pretty complicated. So let's take a look around. There's more. There are other areas to this tumor that look strikingly different. So here you have an area of nuclei that is densely packed. Lots of nuclei all crammed together. It looks much higher grade than the stuff that we saw nearby. And these tumor cells are so densely packed and have so little cytoplasm and look basically like an undifferentiated tumor cell, it makes you think maybe this is an embryonal neoplasm, okay? Something that may have been called a primitive neuroectodermal tumor in the past. So let's look around at our array data and get some better information here. So we see here on chromosome 2 that we've got an amplification of NMYC. And on chromosome 11, we have the picture. This is the cytogenomic microarray picture of Chromothripsis, okay? Multiple double-stranded breaks producing gains and losses, particularly across this area of 11q. So basically the picture here is that 11q blew up, okay? And it's been stitched together back again like Frankenstein's monster. And in fact, it's gained an amplicon as well. And this is an amplicon in CCND1 or cyclin D1. And so we have an idea here that maybe this Chromothripsis event is what triggered the embryonal neoplasm, or what went along with the development of the embryonal neoplasm. And maybe the Chromothripsis event is what also was associated with the gain in cyclin D1. So we did some immunostains for cyclin D1, and this is the interface area between the infiltrating glioma component over here and the embryonal component over here, and you can see that cyclin D1 is strikingly overexpressed in the area of the embryonal component and negative in the infiltrating glioma. So the whole thing was IDH mutant. The whole thing had an ATRX loss and appeared P53 mutant. But we can see that cyclin D1 appears overexpressed in the embryonal component. So our integrated diagnosis here was that we see, at that time, this was diagnosed prior to the WHO 2016, so we used the term primitive neuroectodermal tumor, WHO grade 4, arising from an IDH mutant astrocytoma. And our implications, the diagnosis and poor prognosis in this case is reinforced by the genomic studies. We have a pretty clear idea of that this is not going to do well, unfortunately. Our combined analysis offers some greater insight into the potential mechanisms and the drivers of tumor progression in this case. And the genomics really explain the histology and vice versa. That NMYC amplicon and the cyclin D1 amplicon we usually wouldn't see in a lower grade glioma. That gives you an idea that, oh my, well, I'm dealing with something worse. And then you look at the histology and say, oh, well, that's totally something worse. So you can kind of come from either direction and say that this is going to be an aggressive tumor. And so rarities like that really grant some opportunities to bolster our shared understanding of tumor biology. What is it that causes that tumor to develop a Chromothripsis event and some amplicons in unusual oncogenes that may make it behave in a worse fashion? Our third case, this is a woman in her early 20s who presented us with headaches. It's a common theme here. And she was found to have a solid and cystic left temporal mass. And so this is a low-power view that really gives you an impression of an infiltrating glioma. Okay, so over here, this less involved brain shows low cellularity. But you can see over the course of this slide, okay, we can see an increased cellularity over to here. And so this is a picture of infiltration, all right? These single cells are infiltrating into the surrounding brain parenchyma, and they're doing so singly, which allows you to get this gradient of cellularity over space. So we have an infiltrating glioma, and we do an oncoscan, and it really looks kind of weird. We really don't have any copy number abnormalities here, but what we did have was a mutation in BRAF, in particular a V600E mutation. So a couple of things don't really fit here. So technically this is an IDH wild type tumor. It doesn't have an IDH mutation, but it doesn't really fit into the traditional IDH mutant or IDH wild type tumor in that it doesn't, it really is quite cytogenomically quiescent and just has this BRAF mutation. So we were flummoxed on this one when we first ran into it. We called it diffuse astrocytoma, WHO grade 2, IDH wild type. We were, at first the histology and genomic profile seemed a little bit discordant. We were really expecting this to have an IDH mutation. Instead, we ran into this BRAF mutation. And we considered an infiltrative variant of a pilocytic astrocytoma, but this tumor didn't really have any of the histologic features of a pilocytic astrocytoma, so we ended up just sticking with diffuse astrocytoma grade 2. And importantly, the BRAF mutation does raise the possibility of targeted therapy in this case. So we kept reading, and we ran into other groups describing BRAF V600E mutations in a subset of low-grade diffusely infiltrating gliomas in adults, as stated here. And some of these tumor groups actually didn't behave all that well. They ended up progressing. And our tumor actually did progress in a relatively short period of time. So we're working on seeing if this patient can be placed on some targeted therapy. But further study of the literature, as I said, revealed an emergent group of BRAF mutant infiltrating gliomas carrying some risk of progression. And this tumor did, in fact, show clinical and pathologic progression within a year's time. And I don't think the findings in this case would have been fully appreciated without a wide net of testing. You would have to test this for BRAF in order to find it, instead of just testing for IDH or just testing for 1p19q, say, by FISH. It wouldn't have given you the clearest idea of what this is and what it might do. So this, of course, brings up the idea, how many other tumors have we seen like this? Are these precursors to BRAF mutant GBMs? There are subsets of GBMs. Many of them are epithelioid, and there's some other subtypes that have been described to have BRAF mutations. So we're wondering whether this is a precursor to that or whether it's something different entirely. So our fourth case here is a man in his mid-40s. He has a history of medulloblastoma that he developed as an adult, actually about 10 years ago. And this is over time he's been followed with serial imaging. He had seen marked regression of his medulloblastoma after resection, chemotherapy, and radiation. So he's been followed with serial imaging, which noted this small mass growing in the resection bed of the previous medulloblastoma, but very slowly growing and really not showing the imaging features of a medulloblastoma. So the neurosurgeons were really curious about what this was, what was going on. So they went in and did a biopsy, and what they saw was really quite bizarre. So here we have a picture of the lesion, and we see here we have some ganglion cell neurons here, okay, that are sort of spread about willy-nilly. They're not really well organized here, but they're quite mature looking. And then we have some cells that are small and dark, like these guys, okay, and those are looking a lot like granular cell neurons from the cerebellar cortex, okay? And then you have some larger dark cells here that look more atypical and are a little bit more concerning. Maybe infiltrating astrocytoma, but it's an odd picture overall because the whole architecture is messed up of this. It doesn't look like normal cerebellum. It's just a bunch of cells, some of which you might see in normal cerebellum and some of which look more neoplastic, that are just thrown together, okay? So we went back and looked at the original medulloblastoma, and this very much, well, looks like a medulloblastoma. It's very distinct from what we had, though. These are tightly packed cells that are very blue, not showing lots of cytoplasm, look very embryonal or primitive, not very well differentiated, and highly proliferative. And so it would confirm the original diagnosis, but we also figured, well, it really doesn't look like what we have. So we did oncoscans on both of these. And what we found was that the two were basically identical. I know you see there's a little detected mutation over here. That was a low confidence mutation. Wasn't actually likely there. But the rest of the findings look fundamentally identical. So basically, both of the tumors are cytogenetically incredibly similar. And so we have nearly identical microarray data that, despite this drastically different clinical, radiographic, and histopathologic data, suggests a relationship between these two masses. And in fact, if you'll look back on 17, we see that there's a loss of P and a gain of Q, which goes along with an isochromosome 17, which suggests goes along at least with a group 3 or more likely a group 4 tumor. We don't see the findings also we didn't see findings that would have suggested a Wnt or a sonic hedgehog pathway tumor. And so what we basically came up with in this case is that this is a medulloblastoma with extensive maturation. We're seeing mature tissue elements that are derived from a medulloblastoma. And it's probably similar to what's been described in neuroblastomas more commonly in the past, that these tumors can mature and show different clinical pathologic behavior after the maturation phenomenon occurs. So in this case, to our knowledge, this is the first microarray done of this incredibly rare phenomenon in medulloblastoma. It's only been described 6 or 7 times in the literature prior to this, and I don't think anyone's done a microarray on those cases. So what are the implications here? So what determines this dramatically different histologic and behavioral appearances in these two cytogenomically identical lesions? So could it be changes that we're not looking for? Obviously, we're not looking at epigenetic data. It would be nice to look at a methylation profile of these two masses. Could it be some mutation or fusion that's being dropped in maybe the mature lesion? We don't know because we haven't been testing for those. And what's the comparison with a neuroblastoma? Can we really take the experience for maturation in neuroblastomas and apply this to this lesion or not? But really, this case highlights the power of comparison between tumors, even across long periods of time. That original medulloblastoma was about 10 years ago, using FFPE-based broad-spectrum genomic analysis. It's the broad spectrum that really shows how the abnormalities basically match up in between the two tumors. So our fifth case, this is a woman in her early 30s who came with, again, headache and lethargy, and she had a left frontal enhancing mass. And what we see here is densely packed, highly atypical tumor cells, okay, these guys here and here, and they have prominent nucleoli, these little prominent nucleoli, okay? And they also have these rounded eosinophilic inclusions, these pink things, pink blobs in the cytoplasm. And so when you see this type of appearance, number one, it looks very malignant. Number two, these rounded eosinophilic areas make one think about a rhabdoid tumor. And in the CNS, a rhabdoid tumor is an atypical teratoid rhabdoid tumor. Okay, that's what we call them in the CNS. And so our carrier view here, again, shows on this tumor, we see just a few abnormalities, a gain of 8, okay, a PIK3CA mutation on chromosome 3, a little loss on 13, and then a focal loss on chromosome 22, which is where SMARCB1 is located. And we'll look more closely at chromosome 22, and we can see a homozygous loss right at SMARCB1. So the SMARCB1 has just been chopped out of this ATRT. And so our OncoScan findings, again, homozygous deletion at 22q11 locus of SMARCB1. That corresponds to SMARCB1 protein loss detected by immunohistochemistry. And there's an absence of abnormalities suggested with other that might suggest other tumors. And we also see a few other abnormalities detected by the OncoScan. Our integrated diagnosis was atypical teratoid rhabdoid tumor, WHO grade 4. And this patient had been noted previously to have a low-grade glioma in around this location. So we took a look around in the tissue that we got in this case, and so there's some more. So in the surrounding cortex, we saw neurons, okay, some of which had this sort of fibrillar cytoplasm that suggested maybe that they had neurofibrillary tangles in them. And then there were extra cells added on top, these guys, okay, that looked pretty banal, but looked more like an infiltrating glioma, okay? And these neurons sort of scattered around may bring up the idea of a ganglioglioma, but this was actually cortical brain tissue, and was we didn't feel convinced that this was actually a ganglioglioma. And what was most interesting about it was that by immunohistochemistry, these small tumor cells here had lost SMARCB1. So we actually did an OncoScan on that component as well. And we see here, basically there are no real abnormalities outside of chromosome 22. And what we saw on 22 was a loss, a hemizygous loss, not the homozygous loss that we saw in the true ATRT, but a hemizygous loss in the region of SMARCB1. So our low-grade tumor shows a hemizygous deletion at that locus, and then an absence of other abnormalities seen in the adjacent ATRT. And to us, this suggested that this lesion may be a precursor lesion to the ATRT. And so our integrated final diagnosis was atypical teratoid rhabdoid tumor, WHO grade 4, arising from a lower grade SMARCB1 deficient neoplasm. And so the implications here, though some others have reported ATRTs arising from histologically lower grade neoplasm, some people have said that there are gangliogliomas that have been seen in association with ATRTs and some other tumors as well. We were able to demonstrate a cytogenomic relationship between these tumors using our array. And then we also showed that there was evidence of cytogenetic progression based upon the additional abnormalities seen in the array on the ATRT. to go along with the histopathologic changes that showed that the ATRT was a high-grade lesion, and the tumor next door was relatively low-grade. The proliferative indices were markedly different between the two. It was quite clear that the ATRT was demonstrably different than the lower-grade tumor. But this is another example of some of the explicative power of broad-spectrum testing on histologically distinguishable and separable neoplasms. You wouldn't really be able to do this very well without histology being able to tell you distinctly these are different tumors, and then to be able to go in and do testing on the two separate areas. So in summary, the advent of genomic testing in brain tumors has already led to a marked change in our diagnostic approach. We have gotten to the point in neuropathology here at Emory that we oftentimes end up waiting on the genomic testing results before we make a final diagnosis on a fair number of our cases. Some cases are things like glioblastomas in a traditional clinical pathologic scenario with radiographic data to back you up and histology that's very straightforward. We usually will make that diagnosis without having to wait. But lower grade gliomas, we almost always wait on the genomic data. And then a number of other tumor types, we may end up waiting on genomics as well. So in talking with other folks, many large centers are performing broad-spectrum testing on most tumors detected or treated, and some smaller centers, I can understand there's going to be some challenges there, grappling with the need to address this information while also operating in an environment where money just doesn't fall off trees. So and then also clinicians are going to have varying levels of interest or exposure to this data. And so it's going to put a lot of impetus on pathologists to try to drive some of these changes by following the new WHO diagnostic paradigm. I have some experience in working in a smaller center myself. I don't just do neuropathology here at Emory. I do general surgical pathology at a large public hospital in downtown Atlanta, Grady Memorial Hospital. And so I end up having to make decisions on, in cases where the patient maybe doesn't have insurance or there are much greater cost concerns where I have to make decisions about what testing am I going to choose, which am I going to actually run an array on. I'm much more careful when I'm at Grady, whereas at Emory, we're in an academic environment are much more aggressive in testing a lot of our tumors. So I would say that when you're at a smaller center, it's important to note the necessary components, what I view as necessary components for implementing this type of testing. I do think it's important to have something along the lines of a consensus conference where folks can get on the same page about what this data means for the tumor types. It's going to be very hard to just take the information from, say, an outside center and completely integrate it with what you're seeing histologically. That may be difficult. And then really making sure that the correct information gets transferred on to the treating clinicians. It's just important to have a mechanism for everybody to kind of go over this and to really delve into some of the pre-analytic problems that can lead to difficult or impossible to interpret data. So in general, broad-spectrum testing opens up numerous avenues for improved diagnostic and investigative capabilities, really opens up your eyes as to what's possible and some opportunities to get a better idea of what drives tumor behavior and biology, highlights features that wouldn't be readily appreciable by histologic analysis. I like to say it answers questions that you would anticipate as well as questions that you may not anticipate. So just like I said, I showed with that BRAF mutant infiltrating glioma, in many cases you would have just anticipated to ask the question of, is this IDH mutant or not, and is there maybe a 1p19q co-deletion or not? But in that instance, doing some more broad spectrum testing not just answered those questions, but answered the question of, is there a BRAF mutation, which you may not have thought to ask. So really, this type of testing alters our understanding of tumors past, present, and future. Really makes it very different in terms of interpreting data from the past, interpreting data that perhaps that on patients that are referred to you from outside institutions, and makes you think about differences of how we are going to approach these tumors in the future. And this type of testing is likely to only gain in importance, in my opinion. So we couldn't do this without the support of a lot of different people. I was basically trained in doing this type of testing by these two individuals, Deb Sachs and Mike Rossi, here at Emory. And then these are some of the primary technologists who do this testing. I'm leaving off numerous members of our oncology cytogenetics lab who are integral in making this testing a reality. And to those folks, I offer my apologies. It's not because I'm being mean. And then folks in neurosurgery who have been very supportive of us, and then my fellow neuropathologists here at Emory who have trained me and are extremely helpful in making this testing a reality. And then folks in molecular pathology, neuro-oncology, and our pathology informatics group who also help us out a tremendous amount. So at that, I'll thank you again very much for inviting me to give this presentation. I hope this was a beneficial experience for you, and I apologize for the technical difficulties and the fact that I am a disembodied voice. But I'm certainly more than willing to answer your questions at this point, and if you do need to get in touch with me, I'm actually on the Emory website. You can email me as well. Okay? So with that, I'll turn things back over to our moderator.
Well, thank you so very much, Dr. Neill, and thank you very much for your presentation and bringing forth these case studies today. A quick reminder to our audience on how to submit questions. Simply type your question found by clicking on the green Q&A button at the lower left of the presentation window. Dr. Neill will answer as many questions today as time permits. So Dr. Neill, we do have a two-part question to kind of kick us off today. You mentioned a comparison with a 10-year-old FFPE sample was successful. And what is the oldest FFP sample that they've analyzed, and is there any concern about sample degradation?
So that's a very good question, and if I remember correctly, we've gone back 10 to 15 years and gotten good FFPE data. Now, since we're doing this in from a clinical standpoint, we basically attempt it whenever there's a clinical question that may be answered by this type of testing. So if we were pursuing this from a more translational perspective, we would be testing, of course, tumors that go back much, much further, 20, 30 years. And to my knowledge, folks have had success in doing that for older FFPE blocks. But for us doing this clinically, it actually has been only 10 to 15 years that we've tested up to this point.
All right, well, thank you so very much. The OncoScan potentially can pick up a lot of CNVs, such as homogeneous deletions and amplifications. What criteria do you use to select the clinically significant ones?
Another very good question. So first things first, you need to have criteria for what do you report in general. So for us in-house, we don't report LOH events that are less than 5 megabases, and unless we can be sure that, for instance, that LOH event is surrounding maybe a gene or a locus that is known to be incredibly important in that tumor type. But most of the time, LOH events of under 5 megabases we wouldn't report. And then we also, again, say you're looking at a leukemia specimen, or where you may be interested in Icaros, the Icaros locus. There's only going to be a few probes that you would be able to analyze. So in that sense, you would have to be very careful using just a few probes to try to determine whether there's a deletion or not. But in general, I would state that the answer to your question is the best way to figure out whether something is clinically significant or not is to have the type of consensus conference that I'm speaking of, where really you can get multiple different people in the room, where some folks are coming from neuropathology, some folks maybe from neuro-oncology, and then oncology cytogeneticists and molecular pathologists who will bring a wealth of information about what really is the significant findings in these tumor types, and really what's going to feed back into the diagnosis or prognosis. So it's not really an easy answer, but I think it's important to make sure as many people, as many brains as possible are applied to this information, because it is very complex and it can be difficult to figure out what's significant or not.
Thank you. And our next question is, does the tumor heterogeneity pose a major obstacle in characterizing these infiltrating gliomes?
So yes and no. So tumor heterogeneity in terms of areas of necrosis could certainly produce a lot of difficulty in characterizing these gliomas. However, instances where you're dealing with maybe an IDH mutant tumor, where your main question is there a 1p19q co-deletion or not? The majority of those tumor cells are going to show a 1p19q co-deletion, if not all of them. I know there are actually some folks I know at Brigham and Women's who are doing some single-cell RNA-based testing on gliomas who can tell you, oh, there's a lot of heterogeneity in all of these things. But from a clinical standpoint, we really haven't had a whole lot of difficulty in tumor heterogeneity posing an obstacle to getting us a usable data.
All right, well, thank you so very much. And our next question is, do you report long stretches of the homoglossity if this includes a cancer gene?
So similar to what I said before, our cutoff is 5 megabases, and we basically do that. It's based upon the idea that there will be some homozygosity in general. We also, the testing platform is probably least specific in dealing with areas of homozygosity. So, but to answer your primary question, yes, if there's a long stretch of homozygosity that includes a cancer gene, we would report that. No questions asked.
Oh, great. Thank you so very much here. Our next question is why do you use 5 MB as the cutoff?
So I answered that a little bit in the previous question, but some places use 10 megabases, but in our experience, there are going to be areas of homozygosity that the platform picks up, particularly around the ends of chromosomes and around centromeres, that are basically just noise in the system, and as far as we can tell. So we use 5 megabases just as our way. It is relatively arbitrary, but just a way for us not to be reporting lots of small areas of homozygosity that may not be real or may not be clinically relevant.
Right. No, thank you so very much. And we have time for a couple more questions here. And do you include the Cairo view, the case image, in your final report?
We don't currently put images into the final report. The way our final report is structured is that we basically have an interpretive component. We have a free text area where we try to give an interpretation of the data overall, highlighting certain things and give an idea of what's clinically relevant. We also produce a table listing all of the areas that have been detected as aberrant and that we have agreed are clinically relevant. And then we give cytogenomic nomenclature as well, and then a bunch of disclaimers and so forth.
All right, thank you so very much here. And our final question here for today is, can you list all the diagnosis again? I didn't catch everything.
Sure. So I believe the first one is an IDH mutant infiltrating astrocytoma. The second one is an IDH mutant infiltrating astrocytoma with a what was called at the time a primitive neuroectodermal tumor, now would be called a CNS embryonal tumor or CNS embryonal neoplasm. And then third, let's see, there was a BRAF mutant infiltrating glioma. Fourth was a medulloblastoma with extensive maturation. And fifth is an ATRT arising from a lower grade SMARCD1 deficient neoplasm.
All right. Well, thank you, Dr. Neill, once again for answering the audience's questions here. Do you have any final remarks as we wrap up here today?
Well, I really appreciate the questions, and I had a great time doing this. And please, if you need to ask me any questions further on down the line, feel free to do so. I appreciate everybody's attention and showing up today to hear me. Thank you.
All right, thank you very much. And I'd like to thank the audience for joining us today for their interesting questions. Questions we do not have time for today will be addressed by the speaker after the presentation. We'd like to thank our sponsor as well, Affymatrix, for underwriting today's educational webcast. This webcast will be able to be viewed on demand through March 29th of 2017. LabRoots will alert you via email when it is available for replay. And we encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.